Hello, this is John Rauch, a scientist at Sartorius. Today I'll be discussing a poster previously presented at the Society for Neuroscience conference, titled Quantitative Live Cell Kinetic Analysis of Microglia Function and Morphology. Microglia are the resident immune cells of the central nervous system and play significant roles in the regulation of homeostasis and the management of tissue response to inflammation or pathological insults. Microglia impact synaptic remodeling and turnover of dendritic spines through the removal of damaged or unnecessary neurons or synapses. Limited tools and in vitro model systems exist to enable optimizing, monitoring, and analyzing functional and morphological changes of these cells. In this poster, we characterize rat primary, iPSC-derived, and immortalized microglia, and present data evaluating the ability of these cells to phagocytose pHrodo-labeled E. coli bioparticles and apoptotic neuro 2A cells using a quantitative live cell imaging approach with the Incucyte S3 live cell analysis system for neuroscience. The Incucyte S3 live cell analysis system for neuroscience is a fully automated phase contrast and two-color fluorescent system that resides within a standard cell culture incubator for optimal cell viability. It's designed to scan plates and flasks repeatedly over time. In combination with our software that offers fast, flexible, and powerful control for continuous live cell analysis, comprising image acquisition, processing, and data visualization, all from the comfort of your own desk. Along with the instrument and software, we also offer a suite of reagents and consumables that are non-perturbing cell labeling and reporting reagents. Phagocytosis is the process by which certain living cells ingest or engulf other cells or particles. It's very important defense mechanism against infection by microorganisms. More specifically, efferocytosis is the process by which apoptotic cells are removed by phagocytic cells such as microglia. During efferocytosis, the cell membrane of the phagocytic cells engulfs the apoptotic cell, forming a large fluid-filled vesicle containing the dead cell. In this schematic, we're showing you the apoptotic cell is the smaller cell, and it has been labeled by our Incucyte pHrodo cell labeling kit. Once the apoptotic cell is labeled, at the neutral pH of the cytoplasm, there is no fluorescence. After it is engulfed within the phagocytic cell and enters the phagosome, the pH is then lower at about 4.5 to 5.5. This leads to increased photofluorescence. So essentially, once the target cell is in the cytoplasm, you're seeing no fluorescence, and then after it is engulfed is when you see the fluorescent signal. On the right image panels here, you can see a time-lapse visualization of iPSC-derived microglia engulfing a pHrodo orange labeled apoptotic neuro 2A cell. You can see in the first image, when the cell has not been phagocytosed yet, you see very little fluorescence. In the middle, you can see it beginning to be engulfed by the microglia cell. And then finally, in the third image, you can see it enter the low pH phagosome, and you see that fluorescent orange signal. Next, we'll look at primary microglial efferocytosis of apoptotic neuro 2A cells. Neuro 2A cells were pre-treated with staurosporine for 24 hours and then labeled with the Incucyte pHrodo orange cell labeling kit. Then they were added to pre-plated primary rat microglia. The neuro 2A cells alone, as you can see in the image on the left, have minimal fluorescence, as we would expect. Engulfment of these labeled apoptotic neuro 2A cells by the microglia causes an increase in orange fluorescence, as you can see in the middle image. This fluorescence is then segmented using the automated Incucyte live cell analysis software. The pink you're seeing there is the actual fluorescent, the segmentation mask being applied by the software to those fluorescent images. Using that segmentation mask, we can then quantify the total orange fluorescent area, which is what you're seeing in the kinetic graph on the right. You can see that as we increase the apoptotic neuro 2A density, we see an increase in that orange fluorescent response, as you would expect. Next, we look at characterization of immortalized microglial efferocytosis. So now, instead of using primary microglia or iPSC-derived microglia, these are immortalized microglia cell lines. Now, when using these cell lines, there could be a lot of variability in functional activity, so it's important to characterize these. In this case, in the Incucyte, we're looking at their efferocytosis potential. So the potential for them to actually ingest these apoptotic neuro 2A cells, which is something we would expect a microglia cell to be able to do. The kinetic efferocytosis response of neuro 2A cells, we looked at the response for 3 different immortalized microglia lines, in this case, HMC3, BV2, and C8B4 cells. BV2 and C8B4 cells showed an increasing signal over time, while HMC3 cells displayed minimal efferocytosis. Microplate graph, which is on the top right, shows that kinetic efferocytosis response from a full 96-well plate. So the 3 cell lines were seated on the same plate at 5,000 cells per well, and then pHrodo-labeled apoptotic neuro 2A cells were added at decreasing densities from left to right. Cytochalasin D was added at each neuro 2A density to inhibit efferocytosis. So you can see once again that we're seeing a very little response from the HMC3 cells at any of those densities compared to the response of the increase in orange fluorescence we're seeing from the BV2 cells and the C8B4 cells. The neuro 2A cells alone that are in the bottom 2 rows of that microplate graph show no fluorescent response, as you would expect. This is also supported by the kinetic graph on the top left, where again you can see the response from the BV2 cells and the C8B4 cells over about 20 hours. We're seeing a nice increase in fluorescent orange area and very little increase in those HMC3 cells compared to the neuro 2A alone controls. Images taken at 20 hours, which are in the bottom left, show the fluorescent signal from these BV2 and C8B4 cells and the lack of signal from HMC3 cells over control apoptotic neuro 2A cells alone. So here we're seeing that not only are we quantifying this by looking at the orange fluorescent area, but we're also able to look at these images and visually confirm that yes, we are seeing efferocytosis from the BV2 and C8B4 cells, and visually we can see that orange fluorescence. In the HMC3 cells, you're seeing very little fluorescence compared to that image of the neuro 2A cells alone. Next, we'll look at the effect of cytochalasin D and selengitide on microglial function. So here, we're separating the process of efferocytosis and phagocytosis. To look at efferocytosis, we're once again using the pHrodo-labeled neuro 2A cells. To look at phagocytosis, we're now using pHrodo-labeled E. coli bioparticles. So BV2 cells, which are the effector cells, efferocytose apoptotic neuro 2A cells, which is what you're looking at in the left-hand panels, or E. coli bioparticles, which is in the middle panels. Cytochalasin D, across the top, elicits a concentration-dependent inhibition of both efferocytosis and phagocytosis, with EC50 values of 0.16 and 1.5 micromolar, respectively. Selengitide, an integrin inhibitor, selectively attenuates efferocytosis with minimal effect on phagocytosis at the highest concentration tested of 100 micromolar. These data support the role of integrins in the cell interactions required for efferocytosis, but not the phagocytosis of bacteria-based bioparticles. So this figure accentuates the importance of separating these two processes in terms of using different target material. We can then add a compound, in this case selengitide, that can specifically target one or the other pathway. Next, we'll look at iPSC-derived microglia in terms of assessing their ability to efferocytose apoptotic neuro 2A cells or also phagocytose other physiologically relevant target material. In this case, we're looking at kinetic graphs displaying the concentration-dependent response to either pHrodo-labeled apoptotic neuro 2A cells, beta amyloid, alpha-synuclein, and myelin basic protein. So we can use the Incucyte pHrodo reagent to not only label the apoptotic cells, but also label this other relevant target material. In this case, as I said, beta amyloid, alpha-synuclein, and myelin basic protein. All of these things are shown to have been cleared by microglia. So it's important not only to look at their ability to clear these dying neurons, but also to look at this other protein material. Now we'll be looking at the effect of LPS activation on microglial cell line function. As I stated before, these are two distinct processes, and here we're showing a differential effect of LPS activation. BV2 cells were incubated with 300 nanograms per mil LPS for 24 hours prior to the addition of either Incucyte pHrodo labeled neuro2A cells or bioparticles to induce microglial activation. LPS treatment decreased the efferocytosis response to apoptotic neuro2A cells and minimally increased phagocytosis of bioparticles. So looking at these two distinct processes, we're able to add LPS and see a different response between both of them. Now we'll look at the other benefit of the Incucyte live cell imaging system, which is looking at the images of the live cells. So here, we're looking at a variety of different microglial cell lines, and in testing these, one of the things that was striking was the vast difference in morphology that we saw in these lines. Now, you would expect that these are all microglial lines in some way or another, that they would have similar morphologies. However, that was not the case. So here we're looking at representative images from rat primary microglia on the top left, two different iPSC-derived microglia in the top middle and the top right, coming from Axol or CDI. On the bottom, we're looking at BV2 cells and C8B4 cells, two immortalized murine microglia cell lines, and also HMC3 human immortalized microglia on the bottom right. We can see the differential morphology that was observed depending on the species and the source. So even in the two iPSC-derived microglia cell lines, we're seeing a vast difference in the morphology there. You can see in the Axol iPSC-derived cells, they're much more spread out and flattened, and in the CDI iPSC-derived microglia were much smaller. So, in conclusion, microglia in vitro can have vastly different morphology depending on the species and source of the cells. Microglia can phagocytose a variety of targets, including E. coli bioparticles, apoptotic neuro2A cells, and physiologically relevant protein targets. Immortalized microglia have varying degrees of phagocytic potential, and this can be modified by activation with LPS. Phagocytosis and efferocytosis are distinct processes that can be selectively inhibited by pharmacological means. The Incucyte S3 live cell analysis system for neuroscience can be used with pHrodo Orange cell labeling kit to characterize the morphology and quantify the functional phagocytic and efferocytic activity in these microglia. Thank you.